Hi everyone, my name is Dr. Kenya Fernandez from the University of Sydney, and today I'm going to be talking to you about fighting fungal skin infections with Australian Jarrah honey. So dermatophytes are a group of filamentous fungi, and they cause superficial infections of the hair, skin, and nails. These infections are collectively described as tinnias, and they are the most common fungal infections worldwide. It's been estimated that almost everyone will acquire dermatophytic infection at some point during their lives. And they're also getting a lot more common. Over the last couple of decades, the incidence of tinea of the nails has risen from 2% to 14% in the developed world. And the incidence of athlete's foot has increased to around 20 to 25% of all adults. So there are many factors that have been implicated as being responsible for this, and this includes increased travel, pet ownership, sporting facility use, and an aging population. Currently, around $500 million is spent annually worldwide on antifungal therapies for tinnias. Treatment can be either systemic or topical, with the latter being favoured due to easier self-administration and less severe side effects. But unfortunately, these therapies often take weeks or even months to resolve the infection, and this is with daily or twice daily applications. Relapse is also sadly very common. So the limitations of current antifungal treatments have led to a renewed interest in natural alternatives, and this includes honey. Honey is a promising candidate for the prolonged topical treatment of superficial infections like tinea due to its broad spectrum antimicrobial properties and its low toxicity. Honey has been used in cosmetics and medicines as an antimicrobial emollient and humectant since ancient times, and is still used extensively in a variety of modern cosmetics. Some recent licensing of sterilized honey has also been approved for clinical use. So there are various components of honey which give it antimicrobial properties. This includes high sugar and a low pH. The most significant antimicrobial activity, however, is generally attributed to either the production of hydrogen peroxide or to non-peroxide derived nectar chemicals. So hydrogen peroxide is a powerful oxidant, and it's produced by glucose oxidase, which is a bee-derived enzyme that is activated when the honey is diluted with water. Non-peroxide honeys, on the other hand, originate from manuka plants of the Leptospermum genus, and these contain something called methyl glyoxal. So this is a toxic compound that cross-links and inhibits microbial proteins. However, there are additional non-peroxide floral factors, which can include things like phenolic compounds and peptides that could be from bee or plant origin, as well as other plant factors. So certain unique floral sources from Australia have particularly high levels of antimicrobial activity mediated by hydrogen peroxide, and this could be therapeutically useful for fungal infections. In this study, what we really wanted to do was to characterise the activity of an Australian Jarrah honey against dermatophyte fungi. So to give you some background, the method for testing the activity of honey that we use is determine something called the minimum inhibitory concentration, or MIC. So we use a plate like this full of little wells. From left to right, we have a decreasing concentration of honey, and from top to bottom, each row has a different fungal microorganism in it. We then put this in an incubator to let the microbes grow, and then we look to see what the minimum inhibitory concentration is, which means the lowest amount of honey that could inhibit the growth of the microbe. So you can see these here as these highlighted wells that correspond to the MIC for that particular row. So when we talk about these values, keep in mind that the lower number, the better, because this means that less honey is required for inhibition. Generally, we also test the honey on its own, which gives us the total activity, and then we test the honey after treating it with a catalase solution that abolishes hydrogen peroxide activity, and this gives us the non-peroxide activity. So previous studies have shown that fungi are generally more susceptible to the action of hydrogen peroxide type honeys than to manuka type honeys, and where activity is dependent on methyl glyoxal. Filamentous fungi, including dermatophytes in particular, have also been found to have greater sensitivity to honey than yeast species do. So now I'm going to show you some MICs for some dermatophyte and yeast species, and these have been ranked by their susceptibility to hydrogen peroxide. So the ones at the top of the list are the most sensitive, and the ones at the bottom of the list are the least sensitive. Looking at their susceptibility to 3 hydrogen peroxide-based honeys, we can see that the same pattern holds. The species that are most susceptible to hydrogen peroxide are also most susceptible to the peroxide honeys. And finally, when we treat these honeys with catalase to neutralize the hydrogen peroxide activity, we can see that their activity is totally lost. From this, it's clear that hydrogen peroxide is critical for antifungal activity, but is it the only thing responsible? So our next step was to determine the amount of hydrogen peroxide present in our honey samples. And we did this using a standard colorimetric method. So here you can see the result for one of our Jarrah honeys. And we chose this honey in particular to focus on because it had a high level of antifungal activity, but a relatively low level of hydrogen peroxide activity. So when comparing the MIC of hydrogen peroxide alone to the estimated amount of hydrogen peroxide in the honey samples, you can see that there is about 8 to 15 times less hydrogen peroxide in the honey. So this tells us that while hydrogen peroxide is critical for antifungal activity, it is not sufficient. So this means that there must be something else in the honey that is making it work so well against fungi. So next we wanted to look at the effect of honey on conidial germination. So we took some fungal conidia and we immediately treated them with either honey or hydrogen peroxide for 48 hours. We then stained the fungal cell wall with something called calcifluor white, and then we took a look at them under a fluorescence microscope. So you can see here in the untreated control that the conidia have germinated and produced these long hyphae. However, in all of the hydrogen peroxide or honey treatments, the conidia have been completely prevented from germinating. So this was a really exciting result, as the resilience of these conidia is a big part of why these diseases are so contagious and why relapse is so common. So that's a great result for honey, that it prevents these conidia from germinating. So next, in order to explore how honey actually damages the mature hyphae, we took some conidia and let them grow and mature for 24 hours. And then we treated them with hydrogen peroxide or honey, and we took a look at them under a scanning electron microscope. So treatment with artificial honey and catalase shows that the hyphae are smooth and they have uniform surfaces. Compared to this, however, treatment with Jarrah at either 1 or 2 times the MIC shows prominent bulbs and profusions, with treatment at 2 times MIC showing the hyphae becoming rough, damaged, and collapse. When catalase was added to neutralize the hydrogen peroxide, the hyphal bulging morphology still remained, although some of the roughness and damage was gone. So these hyphal deformities suggest that low concentrations of hydrogen peroxide damage the surface of mature hyphae, but that there are other stressors that may be present in Jarrah honey that cause this ballooning morphology. These results again suggest that hydrogen peroxide production in Jarrah is necessary, but not sufficient for its high antifungal activity, and that there is another synergizing agent present. Potential synergistic compounds that might be present in Jarrah include polyphenols, antimicrobial peptides, Maillard reaction products, and gluconic acid. And finally, we looked at whether and how much oxidative stress honey causes. So levels of hydrogen peroxide that exceed these cellular antioxidant defenses can result in either temporary or permanent oxidative changes to cellular structure, including lipids, proteins, and DNA. So to test whether intracellular reactive oxygen species were generated from honey treatment, we used 2 fluorophores that were sensitive to oxidative species. So Cellrox green and DCFDA. So DCFDA is membrane permeable, and that means that it can be lost from the hyphae over time. Cellrox green, on the other hand, binds irreversibly to the DNA and cannot be lost. So surprisingly, these fluorescence redox dyes were unable to detect internal oxidative stress in mature hyphae following treatment with inhibitory or sub-inhibitory concentrations of honey. Unlike treatment with the hydrogen peroxide control, which displayed significant fluorescence, treatment with honey did not increase fluorescence significantly relative to the untreated control. Antifungal agents that damage and kill cells normally do activate stress response pathways. However, stress pathway downregulation has also been observed following synergistic treatments that seem to disrupt the normal stress response. So together, these results suggest that the antifungal mechanism of honey is largely mediated at the hyphal surface, which is rapidly damaged without invoking intracellular reactive oxygen species. So to summarize our findings, hydrogen peroxide type honey is antifungal, especially against dermatophytes. Hydrogen peroxide is critical for activity, but appears to be powered by additional agents. Honey inhibits spore germination and damages hyphal surfaces, but does not generate internal reactive oxygen species. And all in all, honey activity is complex. Our study indicates that Jarrah honey has unique antifungal attributes that may work to inhibit and kill dermatophytes, and this makes it a potentially promising candidate for the treatment of tinnitus. But of course, we still have some questions remaining. Namely, what powers honey activity, and can we develop peroxide honey as an effective and marketable antifungal? Continuing this project is something that we're currently working on as part of New South Wales Bushfire Industry Recovery Package. Further research aimed at fractionating jarrah honey samples could help identify these potential synergens and enable a potential rapid screening of honey samples for high activity, and in vitro studies to determine whether the potent antifungal activity of jarrah honey actually translate to a mycological cure in a clinical setting is needed. So finally, I would just like to acknowledge the two very talented honours students in our lab, Krish Krishnakumar and Annabelle Gutentag, who did the majority of this work. And thank you very much to everybody for listening. And if you'd like to hear more about our project, you can visit the website shown on this slide.
Hello, everyone. I am very pleased to take part in this conference. My talk is bee products as a promising source of bioactive molecules and functional properties. In the sacred books, honey is mentioned as remedy, and Avicenna in Qanun fi Tib mixed herbs with honey and bee products. Let the primary law of healing, let the food be the medicine. So pathologies promote overproduction of free radicals, reactive oxygen species, and we have enzyme as antioxidants like superoxide dismutase, catalase, non-enzymatic components like vitamin E, C, and E, flavonoids, and other polyphenols. If the oxidants are more than the antioxidants, the body faces oxidative stress. So bee products in cosmetics have a very important effect, and we have honey like moisturizing effect by carbohydrates, amino acids, organic acids. We have also nourishing properties with vitamins, with propolis, antimicrobial, anti-acne, and we have flavonoids, polyphenols as components. With the bee pollen, anti-aging properties, antioxidant effect for protection of skin damage, with sugars, oligopolysaccharides, anti-wicks, and also anti-radiation activity with glycolipids, polyunsaturated fatty acids, anti-aging with vitamin C, vitamin E, vitamin E. So we have also royal jelly as anti-aging, reduces melanogenesis, production of collagen, antiseptic impact, and antioxidant effect by creatine, pinocombrine, proline, cysteine, and sebacic acid. Bee venom, also apitoxin, has anti-aging photoprotective effects by inhibition of MMP1 and MPP2. So to have markers of these antioxidant activities, we have many texts as ORAC, DPPH, ABTS, molybdate, and NO activities with pollen, propolis, and also honey. So in honey, we have minerals, carbohydrates, amino acids, proline, and we have organic acids, vitamins, polyphenols, and flavonoids. So all these components with relationships with these components give us a very good impact on cosmetic uses. Honey for practical uses, we have massage therapy, cataplasm, sit bath, ingestion, inhalation. Honey is one of the first cosmetics in history. It stimulates the production of new cells in the skin and maintains hydration. Acacia honey is the best for this. And in women from the Arab Peninsula, prepare anti-age face masks consisting of honey mixed with egg yolk, avocado, lemon, and yaourt. In Arab medicine, honey is used for fungal infection of the skin, and we mix olive oil, argan oil with honey to cure skin. In Morocco, we have some honey with cosmetic impact, like chamomile honey. We have also euphorbs for skin remedies and as immunostimulatory. Nigella sativa also, honey for allergies, and ruta as anti-inflammatory, Thymus vulgaris as antiseptic antioxidant, and Duplerum as anti-cancer skin disease. So measuring this oxygen radical activity absorbance, we have salvia honey, nigella, origanum, euphorbe, and Thymus honey, ziziphus honey as cosmetic impact of this honey. Also, one marker of this immunostimulating effect is apalbumin 1, representing the most physiologically active component of honey. So honey from Buplorum spinosum and from Ziziphus lotus and citrus have a high level of apalbumins, which have a very good effect, immunostimulatory and antiseptic effects. But also not honey used for external application, but also by oral ingestion. When you make by oral ingestion honey, we have an increase in antioxidant status like vitamin C, beta-carotene, and glutathione reductase. When we mixed honey with the bee wax and olive oil, we have a very good antibacterial effect against growth of Staphylococcus aureus and Candida albinus. It is our evidence-based apitherapy with all honey used as antioxidant, anti-inflammatory, and also with the relationships between biomolecules and physicochemical parameters. I will give an example of Buplorum spinosum honey from Morocco, and this honey it's very rich in minerals like copper, manganese, zinc, and iron, and sugar, fructose, glucose, trehalose, and methylglucose. And when you look at phenolic compounds, we have methyl serengate as good markers and also in a great amount for this honey, like for Manuka honey. And we have a very interesting effect of emergence of multi-drug resistance. But when you look at this honey, we have antibacterial properties produced in the Moroccan Atlas mountain against Escherichia coli, Pseudomonas aeruginosa, and also Staphylococcus aureus. And we have a very good relationship between this antibacterial activity, color, acidity, content of mineraloids, fructose, epicachicine, methyl serengate, comaric acid, and comaric acid type. So this characterization of Xanthas honey is a health-promoting food due to its anti-proliferative and antioxidant activities, should increase its commercial value, which would have a very positive economic impact on the rural communities of Morocco where it is produced. Also, we have the mixture of honey with essential oils. It's named Arho honey. And when we mixed honey with essential oils, we have biomolecules from honey like syringic acid, vanillic acid, caffeic, ferulic acid, benzoic acid. But with essential oils, we have carvacrol, thymol, terpenin, and cineol. And this synergistic effect gives us a very good effect in cosmetic use. And when you look at these molecules, we have these molecules as antioxidants, and it prevents lipid peroxidation, which gives us a very good effect and impact on skin diseases. We have also this honey rich in tannic, ferulic, gallic, epicacin, galate, and minanoid, tends to synergize with the essential oils. In addition, high acidity and conductivity positively influence this synergy. So now I will give you an example of emulsion, but we have many fermented food, pharmaceuticals, and cosmetic contains a lipid phase dispersed in an aqueous medium that they are while in water emulsion. And this lipid phase is constituted by almond and wheat germ oils. And in cosmetic formulation, almond oil has been used in facial care as moisturizer for excessively dry skin, sunburn, and windburn. It is used in lip balms, cream, and in formulation for infants. Wheat germ oil used in microemulsion, lotion, and creams have proven to be effective in treatment of sunburn and in some skin diseases such as psoriasis and eczema. And when you look at this natural antioxidant like propolis and thymus, we have a very good, interesting data with our team developed emulsion in which almond and wheat germ oils were used as dispersed phase, and propolis and thymus extract were used as antioxidants. And the present work deals to compare the antioxidant capacity of this extract in the emulsion as well as their effect on the viscosity of the emulsion. And when you look at chemical compounds of hydroalcoholic extract of propolis and thyme waste extract, We have in thyme extract, we have aromatic acid, benzoic, hydroxybenzoic, comaric acid, and caffeic acid. But in propolis, we have flavonoids, guarantine, pinocombrine, and creatine. So when you look at the effect of adding thyme extract and propolis at different concentration of 0.1, 0.2, 0.04, within its emulsion formulation, we have E, B, C, D, and U. It's 100% with germ oils, and we have 100% with almond oils. And when you look at the level of lipid peroxidation, TBAR and hydroxyperoxide, we have a very good effect on propolis. It decreases this lipid peroxidation with propolis and also with thymus, but the level is corresponding to the dose of 0.02 and 0.04, and we have apparent viscosity decrease with propolis at the level of 0.02 and 0.04. But when you look at 3D surface plot representing this interaction, we have a very impact effect. We have with this the relationship between almond oil and propolis and thymus give us a very increase in propolis, give us a decrease in TBAR and in lipid peroxidation, and the same is correlated with the thymus oil. And we have apparent viscosity is very low when you use a concentration of propolis between 0.03 and 0.02. In order to assess this correlation between this variable, it is clear that TBAR content correlated positively with germ oil content, and we have thyme waste extract was correlated with viscosity and hydroxyperoxide content for parameters which found to be negatively correlated with propolis percentage. So depending on the wise proportion, the emulsion with more significant in propolis was used at the concentration of 0.004%. This finding could be with great value for pharmaceutical and cosmetic industries, and also we present an alternative way to formulate emulsion containing more safe and beneficial products from nature in the intention of replacing synthetic compounds. The use of waste products and recycling then present a profit-making strategy from a lucrative view of our cosmetic industries. Above all, the increase of the use of natural products in emulsion formulation is eco-friendly and could avoid several side effects resulting from synthetic products on human health. And here we give another example with propolis as skin disease, because it is against free radicals and have antibacterial and immunostimulatory effect. And when you look at this propolis on oxidative stability and reactional property of emulsion, we have a very interesting effect, because the analysis of this propolis, we have aromatic acid, caffeic acid, we have phenolic acid ester, like benzene caffeiac, like pentyl coumarat. We have flavonoids, guarantine, pinocombrine, conferol, quercetin, and also alpinol, chalcone. And we have sugar. So when you look at this formulation with this oil, you have protective effect of propolis extract on oxidative stability measuring through peroxide content. And we have a very good effect when you look at this formulation, but the effect is very good with 0.04% with propolis and also with thymus. But in control, we have an increase in lipid peroxidation. The same effect for the oxidative stability measuring TBAR method. We have a very good decrease in TBAR related to concentration of propolis, and the same effect with BHA. But in control, we have an increase in TBAR. So in conclusion, Moroccan propolis used was mainly constituted by flavonoids and phenolic acid. Propolis was better antioxidant in the concentration of 0.02 and 0.04% than BHA in the same concentration in emulsion, while in water, when lipid phase was constituted by almond oil. The ability of both antioxidants decreases with the increase in wheat germ percentage present in the oil in water emulsion. The rheological behavior is typical of a non-Newtonian fluid, more specifically a pseudoplastic fluid in which almond oil is more adequate for having a height stable while in water immersion. And thank you very much for your consideration.
Welcome, everybody. My name is Anna Kurek-Górecka. I am from Poland. The topic of my presentation is evaluation of a photoprotective effect of polymeric gel containing propolis, preliminary study. I would like to start my presentation from describe negative effects of UV radiation. We know that long-term exposure of the skin to ultraviolet radiation is related to premature aging and skin cancers. Among major consequences of UV radiation are immunosuppression, release of reactive oxygen species, release of reactive nitrogen species, and degradation of the extracellular matrix. UV radiation induces immunosuppression and immunotolerance. UV radiation stimulates regulatory T cells, and they are responsible for inhibition of contact hypersensitivity and releasing interleukin 10. The last interleukin 10 is immunosuppressive cytokine. Solar radiation exert negative effects on the skin connective tissue, resulting in the accumulation of abnormal elastic fibers and degradation of collagen fibers, as well as other components of the intracellular matrix. UV radiation increases the number of dysfunctional glycosaminoglycans and proteoglycans. Topical sunscreens based on physical and chemical UV filters are needed but don't provide full protection. So the strategies recommended to reduce tissue damage caused by UV radiation are photoeducation, mechanic photoprotection by clothes and accessories, and topical as well as oral photoprotection. Propolis as a natural substance which exhibit anti-aging, photoprotective, antioxidative, anti-inflammatory and regenerative properties, seems a good agent for photoprotective effect. Phenolic acid and flavonoids are responsible for anti-aging effect of propolis. Among major compounds which are responsible for photoprotective effect of propolis are caffeic acid, paracumaric acid, ferulic acid, quercetin, camphorol, and caffeic acid phenyl ester. Plenty studies indicate that propolis exhibits a photoprotective effect. I would like present result of a study conducted by Butnariu. Nanoemulsion based on aqueous propolis extract and lycopene extract improve skin ability to defend itself from the sunlight. So, aim of our study was to evaluate the photoprotective effect of polymeric gel containing Polish propolis. Our experimental group involved subjects between 18 and 50 years old who applied polymeric gel with ethanol extract of propolis on the square of the dimension 3 on 3 centimeter forearm, as there is no hair. The measurements were of a pilot nature, and 14 volunteers participated in them. The study protocol was approved by the Bioethics Committee of the Silesian Medical University in Katowice, number KNW 0022 KB1 27 I-16. The preparation of hydrogel with propolis was as follows. Ethanol extract of propolis at concentration 50% was mixed with hydrogel based off hydroxyethyl cellulose to obtain polymeric gels at 3 different concentrations, 5%, 10%, and 15%. Mentioned a fat-free base medium has been transparent hydrophilic gel having 3-dimensional structures. It was formed from a semi-synthetic organic macromolecular colloid, hydroxyethyl cellulose, by a solvatation process, and the base was delivered for the study from a company from Krakow. During experiment, we measured skin before and after topical application using a 410 solar directional hemispherical reflectometer. The reflectance measurements of individuals shown the reflectance values which allow concluding that the tested polymeric gel containing propolis protects skin from the access of radiation in the spectral range from 335 to 2050 nanometers. And now I would like to present obtained result. On this chart, you can see the reflectance of the skin without preparation, base and propolis at a concentration 5%, 10%, and 15% at the wavelength at range from 1,700 to 2,500 nanometers. And in this range, wavelength, statistically significant difference in reflectance were shown depending on the formulation applied to the skin. The median reflectance of the skin without the applied preparation was 0.121 and was higher than the median reflectance of the base, for which total reflectance was equal 0.110. Compared to the median base reflectance, the median reflectance of propolis was higher and amounted to 0.122 at the concentration 5%, 0.129 at the concentration 10%, and 0.151 at the concentration 50% respectively. However, in the post hoc test, it was shown that statistically significant differences occurred only between base reflectance and gel containing 50% propolis. And in the table, you can see the value of reflectance for skin without topical application, base and propolis at 3 different concentration. And we measured reflectance at 6 ranges of wavelength. First, from 335 to 380 nanometers. Second, in the range from 100 to 540 nanometers. Next, from 480 nanometers to 600 nanometers. Next, from 590 to 720 nanometers. Next, from 700 to 1100 nanometers. And last, from 1000 to 1700 nanometers. No statistically significant differences in the skin reflectance were found, regardless of the preparation applied to it. And now I would like to present our conclusion. We observed that the median reflectance value had range from 1000 to 1700 nanometer and the range from 700 to 1100 nanometer wavelength was similar to that at 1700 to 2500 nanometers. The median reflectance of the skin without the preparation was higher than the median reflectance of the base, while the median reflectance of propolis were higher than the median base reflectance, demonstrating statistically significant differences and the formation of certain trends at high wavelength values is the basis for further research to increase the number of volunteers participating in the measurements to more precisely determine the effect of propolis radiation on the skin. Propolis exhibits promising photoprotective effect, so it might be used as the potential sunscreen and anti-aging agent for cosmetics. Potentially, it may be incorporated into different cosmetic formulation, and it may be combined with filters and plant extract to improve photoprotective effect. Thank you very much for your attention.
Hello, my name is Thiago Guardia. Welcome to this presentation. Thank you very much for watching it. Today I'll be talking about honeybees, apitherapy, and human longevity. So let's get started with some facts about aging and longevity. Here are these graphs depicting how humans have been, in general, extending lifespan over the past centuries. On the left, we see that in the 1800s, life expectancy was around 45 years old, and on the right, the projections indicate that life expectancy will vary from 60 to almost 90 years old over the next decades. The science of aging and longevity is still evolving, and one of the main references in this field is Dr. Dave Sinclair, which has his brilliant career in the field of biology of aging and translational medicine for aging. His work is dedicated to increase our understanding about what contributes to aging and how to prevent it. According to his projections, next generations will continue to gradually experience an extended lifespan, and many people will be able to live beyond 100 years old with a very good level of health. According to his research, the main cause of aging has to do with epigenetic changes. So basically, changes in the expression of our genes is what is associated with great impact in the physiology and longevity of our physical body. And a very important aspect of epigenetics is that the expression of our genes is greatly impacted by our lifestyle. So this means that the lifestyle choices that we make, so the food we eat, how much we exercise, the quality of our sleep, the environment we live in, among other factors, are in the end of the day what mainly determines how long we are going to live. And of course, when we think about honeybees and longevity, we kind of automatically think about the longevity of the queen honeybee. The biological development of honeybees is such an incredible demonstration of the effects epigenetics and lifestyle factors have over longevity. In this case, the main lifestyle factor involved is diet. So both the worker honeybee and the queen honeybee are female bees that have the same genome. However, during the early development stages, nutrition is what triggers the expression of different genes, which in turn will determine whether the larva of the bee will become a worker honeybee or a queen honeybee. So the same genotype become a very different phenotype due to nutrition. The queen honeybee can live somewhere between 3 to 5 years old, while the worker honeybee lives around 3 to 6 weeks. This means that the queen honeybee can live around 80 times more than a worker honeybee due to nutrition and other lifestyle factors. So the cells of the human body also have the same kind of epigenetic mechanisms. Of course, humans will not suddenly start living 80 times more by just incorporating a healthy lifestyle, because there are differences between species, of course, but the essential epigenetic mechanisms, such as DNA methylation, for example, are quite similar between bees and humans. Thus, the lifestyle we live is determining the expression of our genes, which in turn influence how long and how healthy we will live. Some studies have also demonstrated that royal jelly and its components have improved health span and extended lifespan in different species, and the mechanisms associated with such effects are also starting to be mapped. There are also some studies on the longevity of beekeepers. So there's this popular concept that beekeepers tend to live longer, and this particular study demonstrated that the telomeres of the genes of beekeepers are longer than the ones from non-beekeepers, and that longer telomeres were associated with longer periods of consumption of the products of honeybees. Thus, this study indicates that regular consumption of the products of the beehive might be beneficial to telomere length maintenance, which in turn is associated with longevity. One of the greatest challenges associated with aging and longevity is that as people are living longer, neurodegenerative diseases, cognitive decline, and dementia are spreading worldwide. And this is one of the main health-related challenges humanity are facing right now. Over the past few years, I did have the opportunity to learn and do some research about the effects of aging in the brain and cognition. On this slide, there are some pictures of the article from my master's degree in neuroscience my colleagues and I published about the neuroprotective effects of oral consumption of royal jelly. So very briefly, we found that regular consumption of royal jelly reversed some of the neurodegenerative process associated with aging, in addition to improve memory. I won't get into details here due to time restrictions, but essentially our results and the results from other studies on the same topic support this idea that royal jelly can be considered a very good supplement for brain health and cognitive longevity. In my PhD, I had this opportunity to study the importance of lifestyle factors for preserving brain and cognitive health during the process of aging. And there on the right, there is a kind of homework for those who want to live longer and live better. So according to many references in the field, physical activity, nutrition, sleep, social life, cognitive engagement, and management of both physical and mental health conditions are considered the factors that influence the most both brain and cognitive health, in addition, of course, to support longevity in general. Apitherapy is this ancient and science-based healing practice that has been used to support human health for millenniums. Many ancient civilizations used to approach bees for healing purposes, and there were a variety of practices, rites, ceremonies associated with that. And the development of apitherapy as a practice in modern medicine is also quite interesting as well. And nowadays, there are literally thousands of scientific papers about the therapeutic properties of the products of the beehive. And as you can see, all the products of the beehive have many functional properties that promotes health and recover our health, thus supporting longevity and quality of life. It's also important to highlight that respiratory issues are among the leading causes of death according to the World Health Organization. This data is before the pandemic, and as we know, taking care of our respiratory health has become even more important nowadays. And then there is this incredible technique called beehive air therapy, in which people breathe the air of beehives for therapeutic purposes. Beehive air therapy can be very effective for treating respiratory issues, and that's also one of my topics of research. This is a relatively recent modality of therapy that has been gaining great popularity over the past few years. And this is because this therapy really works. It works really well. Many people are getting really better from their conditions. And last year we had an entire event that was all about this therapy. The event was organized by Dr. Stephanie Stengasu, Betty from Bikura System, and I. We already started to do some investigations on the pharmacology of behavior therapy, and we plan to start publishing materials about it next year. And for the next year, we also are starting to plan an international clinical study to document the efficiency of this therapy. So as I mentioned, many people are getting better from different conditions, many successful anecdotal cases. And now we want to take a step further by doing scientific research, and to publish the results. It's also important to highlight that behavior therapy, the benefits of behavior therapy goes beyond our physical body. When we breathe the air of the beehive, we are not only absorbing that incredible combination of beneficial molecules that are inside the hive that comes from the products of the hive. When we breathe the air of the beehive with the proper intention and technique, we can also absorb a great amount of life force that the bees keep inside the hive. bees are those enlightened beings that are intimately connected to the energy of the sun. The sun is the great source of energy, warmth, and that sustain life in this planet. And bees carry this direct connection to the life force, to the source of life inside the hive. So when we breathe the air of the beehive with this intention of connecting to the source of life, connecting to life force, open to expand our consciousness, the results are strongly expanded. During the last summer, I had the opportunity to do some experiments and do some very special therapeutic sessions in which we explored the applications of different pranayams associated with sound healing as well. And the benefits are greatly expanded. So it's my great goal to develop this combination of behavioral therapy and subtle practices of breathing and pranayams and sound healing, and continue to spread the benefits of this practice. The results were incredible, and please feel free to get in touch if you are interested in knowing more about it. So this is the last slide of my presentation, and this slide itself has materials for a sequence of presentations and can guide the development of different initiatives. But here, very briefly, I just want to mention that we can establish a healing relationship with honeybees from different levels of perceptions, different levels of intentions, different levels of consciousness. And establishing this healing relationship with bees goes beyond just healing our own bodies. It can help us heal how humanity exists on this planet. It can help us heal our relationships with others, our relationship with ourselves, our relationship with our planet, and not only support longevity of our own physical body, but also support the longevity of our species on this planet. So interacting with bees allow us to have a better perception of the natural world, help us establish a deeper connection with nature, and it also helps us establish a deep relationship with our soul. So I thought it was important to conclude this presentation that beyond the longevity of our body, interacting with bees can enlighten our consciousness and support the development and longevity of humanity as a unity. So thank you very much for watching to this presentation, and feel free to get in touch in case you have any questions or are interested in interacting with me about any one of those topics. Here are the main channels you can find me online, and thank you very much. Namaste.
Hello everybody, I'm Dr. Zorica Plavšić. I'm coming from Belgrade, Serbia in Europe, and I wish you all the best during this wonderful meeting. In the first place, I want to thank you, the organizer, for inviting me for this meeting, and I will present a paper, Apitherapy in Oncology. Does it work? Cancer is a global problem. In many countries, together with heart diseases, it is on the first or second place with regard to the leading causes of death. About 7 million patients die of cancer annually, and 25 million people are currently living with a diagnosis of cancer around the globe. Due to the fact that the average age of the general population is increasing, the global burden of cancer is also increasing. An average of 51% of cancer patients use some kind of alternative or complementary medicine. I prefer to say and use complementary medicine because of non-mainstream practice used together with conventional medicine. I also like to say that apitherapy is a part of integrative medicine, because the integrative medicine we can use these B products together with the conventional medicine without any side effects. This amazing small insect is preparing for us 8 different products in their hives. Each product contains bioactive components, 100 of them, which help us to be healthier, to prevent the disease, and to use it as a medicament for treating the diseases. Let's start with the honey. We can say that honey has anti-cancer mechanism and the effects of apoptosis of the cell, mitochondrial pathway, outer membrane permeabilization, anti-inflammatory, immunomodulatory effect, modulation of insulin signaling, and estrogenic activity. In anti-cancer effects, on the first place, the honey can introduction the apoptosis, death of the cells. Some of these biomolecules rise some actions in the cell, some decrease, but all of them are working synergistic, and the end effect is synergistic. Also, the modulation of oxidative stress, anti-inflammatory effects, anti-proliferative effects, immunomodulation effects. All of these keep us healthy, and these biomolecules work against carcinoma in the body through induction of apoptosis on the first place. The second product is propolis. He also has immunomodulatory activity, apoptosis or death of the cancer cells, cell cycle arrest, regulating some oncogenes, anti-angiogenesis, anti-migrative activity to prevent dissemination of cancer cells, anti-proliferative, cytotoxic activity, and anti-inflammatory activity. A group of authors in this study that was published in 2020 find and explain how propolis act against skin, prostate, breast, gastrointestinal, and oral cancer. All these activities of propolis are within the cancer cells or stabilizing the immunity of the body. Bee pollen also has anti-cancerogenic effect through flavonoids and phenolic acids, very active biomolecules from bee pollen. The anti-cancer impact of bee pollen administration is the stimulation of apoptosis, the inhibition of cell proliferation in multiple cell lines, and the reduction of tumor growth in breast and prostate cancer. Oral intake of royal jelly will work through tumor necrosis factor and transforming growth factor. Tumor necrosis factor produce anti-cancer effect, decreases in tumor size, and transforming growth factor slow down the adverse events like anorexia and fatigue. And the authors say that oral intake of royal jelly improves anti-cancer effects and suppresses adverse events of molecular targeted therapy by regulating TNF-alpha and beta in renal cell carcinoma. The effect of the synergistic interaction of royal jelly with anti-cancer drugs on the cancer cells was improved, as well as normal cells. In this paper, Nutrients, in 2022, this year, and these effects are reduced to reduce the cytotoxicity in normal cells and to increase the apoptosis of cancer cells. Bee venom acts through dendritic cells to stimulate the immune system, through which it activates cellular immunity. Its antioxidant activity is associated with a reduction in reactive oxygen species and elevation in antioxidant enzymes, which leads to protection against cell death, protection from apoptosis. Bee venom, the active biomolecule is melittin from bee venom, and is considered cytolytic but non-specific to cell. Melittin has the ability to suppress tumor growth in breast, liver, prostate, and lung cancer cells. In vitro and in vivo studies show that melittin can suppress growth of cancerous cells by inhibiting NF-κB signaling and activating caspase 3 and 9 pathways. Inhibition of hepatocellular carcinoma cell motility was observed in vitro and in vivo by suppression of RAC1 dependent pathway. Next several slides will present the current research on different cancer cells with bee venom, but all these studies are in vitro studies. In the studies on ovarian cancer cells, bee venom caused apoptotic cell death by increasing the expression of that receptors and inhibiting the JAK STT-AT pathway. This pathway plays a central role in the survival and embryological processes and may show abnormal activity due to some genetic mutations, polymorphism, and alter the functioning of the pathway, leading to the development of cancer. So bee venom can be used in the prevention of development of cancers. whether the growth of human cervical tumors was inhibited by bee venom on mice. It was investigated and it was found that tumor growth was inhibited as a result of the inhibition of nuclear factor kappa B by increasing that receptor. This paper was published in 2015. In leukemia, in a study conducted on human leukemia cells in 2020, it was stated that melittin induced apoptosis in leukemia cells and could be a therapeutic agent in the treatment of leukemia. The anti-cancer activity of bee venom has been demonstrated in the human lung cancer cells line by introducing apoptosis and inhibiting the expression of cyclooxygenase in human lung carcinoma cells. It thus seems that bee venom may have an anti-tumor effect of a human lung cancer. Unlike some non-steroid medicaments, bee venom was not observed to inhibit COX-1, and it is possible that it will not incur the serious adverse effects such as peptic ulcer formation and renal dysfunction associated with the COX-1 inhibition. In vitro study conducted in 2018, authors examined the effect of bee venom on colon cancer cells. It was shown that apoptosis was induced and colon cancer cells' growth was inhibited by activation of dead receptors and inhibition of nuclear factor kappa B. In the conclusion, I will say that the level of knowledge of the patients about apitherapy is quite low, only 41%, and they receive the information they gain from their family or friends in 63, almost 64%, not from the doctor. The rate of use of apitherapy products by patient is also low, 27.6%, and the most frequently used product is, as you all know, honey. Healthcare professionals should conduct more scientific research on the use of apitherapy products in cancer treatment and inform patients about use areas of apitherapy. More clinical studies are needed for the development of apitherapy in oncology. Thank you for being here, and I would like to be in touch with all of you, and here are my address, phone number, email, and website. Thank you again.